So today my presentation is mainly about the risk-based approach. How do we use that to control the AMR? And you may notice that I have food and environment as the two different sources that I want to talk about, how we can control from those two exposure routes. And I used a different font size, but it is not a typo. And I meant to do that. Yeah, because as we noticed that it might be from multiple different exposure routes for the human being to expose to the antimicrobial resistant bacteria. However, so far there's a very imbalanced scientific evidence body that you can find focusing on the two different exposure routes. So in my group, we mainly focus on food safety risk assessment, particularly quantitative microbial risk assessment. We use those tools to address the food safety issues and enhance public health. And we also do some work of risk assessment focusing on antimicrobial resistance for both of those exposure routes, through food and through environment. But I didn't mean to provide the findings from the project in my group. And again, from this presentation, I didn't mean to lead you to any conclusions. Is the food route is more important or environmental route is more important? No, that's not the major goal. The major goal is to introduce the tool of risk assessment and introduce and use several examples from the publications to show how risk assessment can help us to combat AMR issues. So that's the main purpose of this presentation. And for myself, currently I'm an assistant professor in the Department of Food Science and Technology. And actually, I feel I have some connections with you guys from Maryland because I used to be a postdoc at the Department of Food Science and Technology. And over there, I work on nutritional risk assessment. Okay. Here's just some numbers. I guess you already know about this to show how serious the problem is. So on the left side, those are the most recent estimation about the AMR-related infections in the United States annually. And it can lead to about more than $55 billion annually. That is due to the healthcare cost and also due to the loss of productivity. And also is indicated this number, it may be likely to be underestimated. But actually the problem is not only within the United States, it's actually threatening the population globally. So you can see on the right side, it's based on the review on antimicrobial resistance. It's estimated by 2050, there are more cases that we can expect. Actually, it's death cases that we can expect associated with antimicrobial resistance by 2050. It's going to up to 10 million. So the purple part is actually the extra cases from now until 2050. You can see when it's add up to the whole numbers, it's even higher than the numbers death due to cancer or some other very pernicious infectious disease. And the total cost could be 66 trillion pounds. Okay, so let's think about where the antimicrobial resistance in bacteria comes from. And first, I want to remind you that the antimicrobial resistance is a natural phenomenon. So it doesn't mean it's always related to the human activities. It actually has been existing in the nature for quite a long while. That's because some bacteria actually can produce antibiotics. That's their way to survive. That's their way to fight against the other bacteria. So that's trigger and provide some low degree selection environment for the antimicrobial resistance. That is nothing about the human activities. But it's generally considered there are two other ways that maybe trigger the development and existence of AMI in bacteria. So one is the use of antimicrobials for human health. So on the right side for this figure, can you see my mouse? Can you see my cursor?
Yeah.
Okay. So this is a chart. It's a little bit of small font size, but In the x-axis, that's the amount of outpatient use of a penicillin among different member states in Europe. And on the y-axis, that is the percentage of case that has been identified as a penicillin resistant with Staphylococcus pneumonia. So you can see there is a very clear positive association between the use of a penicillin and the proportion of the case identified as a penicillin resistance. That indicates a very strong correlation between the use of antibiotic use among human beings and the resistance cases among human beings. In another source is actually main sources that all the media and science has been focused on is the use of antimicrobials among the for animal health purpose. Particularly, there are some medicines has been used for the growth promotion purpose. Those has been critiqued the most. So on the right side, the very right side, the bubble chart, it shows based on the data from FDA, among 41 different antibiotics that has been approved for the animal or livestock use in the United States, 31 of those 41 are identified as medically important for human use. So with that situation, it might compromise the treatment efficacy for if we use the same class of drugs to treat the human disease. Okay, so when we're facing such a series of problems, do we still use precautionary principles? So when people usually say, okay, we need to use the precautionary principles, most of the time is because we don't have enough scientific information to support our decision making. But I think we are already getting started to collect more information, and it's time to transit into a risk-based approach. And also, I want to emphasize that the risk-based approach needs to have a large scientific evidence to support. Okay, so since I mentioned about the risk-based approach, so let's introduce several new terminology. So first is risk analysis. This is a chart that from the Codex Alimentarius Commission. They recommend if we want to use risk analysis as a tool to control the foodborne antimicrobial resistance and what it should be like. So this is a proposed framework. On the left side here, over here, you can see very clearly there are several components that can be considered as a key component for risk analysis. I would say for this one, is actually proposed one additional that is called a preliminary risk management activities. But usually when we talk about the risk analysis is composed of three components. It's a risk management, risk assessment, and risk communication. So when we come back to this specific framework, it starts with the preliminary risk management activities. So in this step, first is to identify the most important AMR food safety issues. So if you have multiple different pairs and combinations of a bacteria in different food and resistant to different antimicrobial classes, which is more important? And which one actually you need to focus more and collect more information and conduct a more detailed risk assessment? That's something needs to be addressed in the preliminary risk management activities. So once you have that identified, and once you have the preliminary risk management goals established, then let's go to a risk assessment for a specific risky pairs that we want to evaluate. So risk assessment is considered as the scientific component that get together. It's relatively independent from the risk manager, but it's based on the goal that we want to achieve, how to manage the risk, is provide some integrated science information to inform the risk management strategies. And of course, after that, based on the findings from a risk assessment and the risk managers start to evaluate, identify different risk management options, and start to do the implementation, and afterwards do the evaluation. So this is a very general framework that is recommended by Codex. How do we use a risk analysis approach to control the AMI? It's actually since I would say 1990s. Risk analysis has been recommended in worldwide as a preferred way to set the food standards, to trigger more homogenized standards and facilitate fair trade in the worldwide. Okay. So let's continue to focus on risk assessment more. So this is a scientific part. And then later in this presentation, for the examples we're gonna share and discuss, is all risk assessment projects. Risk assessment usually include 4 different steps. It starts with hazard identification. So this is to identify what are the hazards that can potentially pose adverse health effects among the population. And followed by that, there will be the exposure assessment. That's to quantify or either qualitatively or quantitatively to describe the frequency and severity. It's a frequency and the amount of the hazard that the population can be exposed to. So on the right side, that's the hazard identification. And some cases it's called the dose response. So based on the title, it's very obvious that it's to build the correlation between the amount of exposure and the probability of getting the adverse health effects. So when you have the exposure, from the exposure assessment, the amount of exposure, put into the dose response model, then you can predict what is the probability of having the adverse health outcome. So that's actually what risk characterization will help you to do, is to integrate all the information from the first 3 steps and to estimate the risk level to determine it is acceptable or not acceptable, and use that to answer the question, what are the potential interventions that we can use and how it's gonna influence the public health. So on the left side, I basically list all the questions that risk assessment can help to address. Okay, so a typical way of doing a risk assessment, especially for a quantitative risk assessment, is to build a mathematical model to consider all the different risk factors along the way, to describe how the different factors can influence the introduction and the dynamic of the hazard of interest, and all the way to the human exposure. But we usually started with a qualitative description like this. It's called conceptual model. So you need to first understand what is the system that you're describing. And this is a chart showing different exposure pathways that can lead to the human infection of antimicrobial resistant bacteria. So in the center, it is summarized as the classical pathway. So you can see from here, food is in the center, but it could be starting from the food animal where the antibiotics can be used. That could be the trigger of the development of antimicrobial resistant bacteria. And from that, the food from of the animal origin can be contaminated. And then through the consumption of the food, and then people can ingest the resistant bacteria and got infection. Or there could be another way, that's from the human being, because we also use antibiotics. We actually, as the major consumer of the antibiotics, antibiotics. And through the cross-infection that the antimicrobial resistant infection can be spreaded among the human being ourselves. So those are the 2 major different source and routes considered as the classical pathways. But there's some other alternative pathways that can be considered. So for example, there's animal waste from the food animal primary production or slaughterhouse facility. And from that can contaminate the environment. It could contaminate the soil, it could contaminate the water, and all the way to some crops in the field. And as another type of food that can contaminate can get the human being infected. Or here, there's another way that's from the pet animal or companion animals, and they can use antibiotics that are relevant to the human clinical settings. Then through the direct contact, then they also can infect the human being. So here is considered as some alternative pathways. But there is another pathway here is called reverse pathway. So this means, okay, it's not only always start from the food animals. It's not only the direction like from the food animal to food or to human being. It could be reversed. It could be starting from the humans. We, among the human communities, the resistant bacteria has been developed and it can be transmitted through the waste and have the impact on the food animals. And to come back to the circle. So I will say this is a relatively complete picture about the different sources and the possible transmission pathways. However, if you look at the literature, we have a very imbalanced focus. Most of the publication is about starting from the antibiotic use in livestock and how it's going to have the impact through the food to the human being. And maybe there are some evidence over here, but in the media, the way or the weight they have been emphasized is totally different. And there are some studies or researchers, they start to step into this alternative pathways. But so far, most of the application is still within for pathogens, not really resistant pathogens or resistant indicator bacteria. But you can imagine that we're more ready to go into this route. However, for the reverse pathway, I have to say, we almost have none in the risk assessment studies consider the directionality problems. So it's always have a pre-assumed assumption is the direction is always from food, but actually it may not be the case, but that's something that we need to consider. Okay, so as I showed you that it is a very complex network systems. However, most of the evidence that I can show you today is focusing on the food pathway. So I summarized several questions based on current application of a risk assessment for controlling AMR, and what are the questions that have been used risk assessment to answer. So the first one is, what are the most important pairs of AMR bacteria and food? So if you still remember the chart from the Codex recommendation of the framework, it is more like building a risk profile and identify the most important risky pairs and where we should more efforts to continue to investigation. So it is a preliminary risk management activity. So once we have the most risky pairs identified, or the most risky maybe antimicrobial resistant bacteria identified, then people may do some comparative analysis to see, okay, it may be from different sources of food and which food is more important. If I can identify which food is the major source of that specific resistant bacteria, then we can put more efforts to control the contamination in that food, processing the production chain. So if we have a more specific focus identified for a specific antimicrobial resistant bacteria in a specific food, then we can trigger a more detailed risk assessment to answer the question, okay, let's evaluate what is the current risk of exposure to that bacteria through that food. It is okay, and how high it is acceptable, should we further reduce? And using the model, we can also evaluate, okay, if we have some interventions, for example, to withdraw the antibiotic use in livestock, will it have a great impact on the public health? Will it be an effective way to control our concern? So, and also there are some other control measures can be evaluated. So all of those 3 questions at the bottom is usually can be addressed by a more detailed quantitative microbial risk assessment. So in the following slides, I will use several examples to show for each of the questions how risk assessment can do the job. Okay, so first the question, what are most important pairs of antimicrobial resistant bacteria in food? So this approach is usually called risk ranking. And this is a publication that you can use a good reference to study this approach. Excuse me. So this study is the risk ranking of antimicrobial resistant hazards found in meat in Switzerland. So this is their conceptual model. As I mentioned, that they always have a conceptual model to give you a roadmap about what is happening in the whole project. So this is a study conducted in Switzerland. At the very beginning, we have 208 different combinations of bacteria species and food and 2 different antimicrobial class they want evaluated. So basically, they make the comparison across all the 208 combinations. So I have to mention that this is a semi-quantitative analysis, which means they don't necessarily provide for each of the combination what is the risk level. It's not quantified, but they give you the score for the exposure assessment and also hazard characterization for each of the pair. So using the multi-criteria decision analysis approach, they integrated the scores from exposure assessment and from the hazard characterization to give a final index to indicate the risky level for each of the pair. So using this information, they can inform or give some preliminary information for prioritize the risk mitigation activity. Okay, so here's some results from this study. So they started from 208 combinations, but at the end, several tests were excluded because it has been shown in the first step of the chain is always a 0. So there's no way to include those pairs. So they end up have 145 combinations being ranked. So 37 from beef and 46 from pork, etc. And on the top, it is the summary of the top 10 combinations of animal species and bacteria and antimicrobial classes. So you can see all of the top 10 is actually focusing on poultry. Indicate poultry product is really a risky product, at least based on the data from Switzerland study. And there's one particular combination here. You can see poultry for ESBL, that is extended spectrum beta-lactamase producing E. coli, and a PMC producing E. coli. They have a high rank for both exposure assessment and also high rank for hazard characterization. So high rank for exposure assessment means it is likely to have the human being exposed to that hazard through the consumption of a poultry product. And the high rank of the hazard characterization means it's likely to trigger higher probability of getting the disease if the exposure happened. So that's what the high rank means. So this combination has been identified, okay, this is probably the high risk pair that needs to be paid more attention. And in addition to that, they also look at 2 different type of bacteria, and Campylobacter and Enterococcus has been identified as a higher rank. So how to read this? So on x-axis are different bacteria species. On the y-axis is their rank number. So you can see total we have 145. So if they have rank 1, which means that is most risky, and here all the way to 145. So this is just a box plot showing the distribution of the rank for different bacteria species. So when the mean actually is lower in the plot, it means it has a higher rank or it is more risky. So here you can read, okay, Campylobacter and Enterococcus is more risky bacteria. And another chart we also summarize, and for different antimicrobial class, so cephalosporin or third generation cephalosporin have the higher rank, and also macrolide and quinoline, or maybe in the class of fluoroquinoline and the tetracycline have the higher rank. So at the end, they make the conclusion that tetracycline and the macrolide-resistant Enterococcus, fluoroquinoline, and the macrolide-resistant Campylobacter also ranked very high. So this study shows doing the risk ranking to help the whole country first have some targets to work on, right? So the next question is, what are the contributions from various foods to human exposure to a specific antimicrobial bacteria? So to answer this question, comparative risk assessment or comparative exposure assessment is a tool can be used. And here's an example. And this is a study of comparative exposure assessment of, again, is ESBL producing E. coli through the meat consumption. And here I want to mention that sometimes we call this risk assessment, and you can see here is called exposure assessment. That's because there's there's a great challenge for us to estimate from the exposure all the way to the risk. When we talk about the risk, that's indicate the probability of having the disease. It's associated with the exposure, but you need to have the dose response model to help to link the exposure to the probability of the disease. However, for the antimicrobial resistant bacteria, so far we don't really have a good progress on quantify the link between exposure and the outcome. So for regular pathogens, for example, like a salmonella, and when we do have the dose response for salmonella, for E. coli 157, for Listeria monocytogenes. Sorry about that. But those are mainly focused on the susceptible bacteria. A lot of questions need to be considered if we can really make the transition from that dose response all the way to the application to the resistant bacteria. Because for resistant bacteria, it could be more complicated process within the host body. For the bacteria carrying the resistant genes, maybe horizontal gene transfer can happen. So even for the bacteria that carry the gene is not pathogenic, but they still have the potential to trigger the resistant infection because that gene can be potentially transferred to the susceptible pathogens that the host already carry. So there are more complicated scenarios that need to be considered. That's the main reason so far we don't have a good dose response model for the resistant bacteria. So when you search in publications, don't be surprised. There is a lot of risk-based approach that actually measure the exposure rather than the real risk of hazard. Okay, so let's come back to this study. This is a comparative exposure assessment of ESBL E. coli through the consumption of meat. So this is a study conducted in Netherlands. And again, the hazard of the focus is ESBL and PMC producing E. coli. So they focus on different animal-derived foods, meat and poultry. And for this model is quantitatively measure the exposure at the end as the model output. So on the right side, you can see the scope of this food chain it has been described. It's starting from the pre-retail processing, including some sorting, drying, fermenting, heating, or there's nothing over there. And after the retail, then the consumers purchase the food and take it back to their home, and there was a storage, and during which the time and the temperature during the storage can influence the dynamic of the population of bacteria of interest, and then start to do the cooking, that's the heating process. Thermal inactivation can be expected. But again, if without good hygiene practice in the kitchen, cross-contamination can be considered. So these are all of the procedures that has been modeled using mathematical modeling approach to describe the introduction and the transmission along the way of their interested bacteria. So at the end, they can have the exposure among the consumers estimated. So using this tool, and this study identifies some products that have leading the higher exposure to EEC. So let's take a look at their results. So this is the main finding here. So for the exposure to EEC, they identified among the Dutch population, actually the beef consumption actually contributes more than chicken, followed by pork and veal and lamb. So 87% of the total exposure among the Dutch population to EEC attributable to beef. And if you look at why beef has actually have such a great contribution, it's mainly because in the Dutch population, they consume what is called the filet American. It is a minced or ground beef that eaten raw. So it's actually that food itself contributes 61% of the exposure, and followed by the chicken fillet. So at the right side, this is a chart to show, based on the model, they can predict at each of the steps along the chain, what is the contamination level in the product. So you can see it doesn't really differ very well, but here I show you, this is a line for the chicken, and this is a line for beef. So at the very beginning, you can see chicken's contamination is actually much higher than beef, all the way to the step of after storage. It's because for chicken product, most of the time, there's a cooking step involved. So at the end, at the time of consumption, the exposure is lower than beef if they eat raw. So that's the main reason that the beef is actually trigger a higher exposure of the EEC among the Dutch population. So you can see this is an approach that if you have the information along the chain, that you can connect all the dots and help you to answer at the end of consumption, which food is actually posing higher risk of exposure for a specific antimicrobial resistant bacteria.
Can I ask you a question about that?
Sure.
So why do they continue to eat raw beef if they know that, I mean, we've obviously gotten the message out about cooking your chicken to a certain degree of doneness, and we know that beef, when it's a whole muscle cut, we can eat it, rare. Well, I can't, I mean, I don't know. I'm the type that when I cook, like I'm usually nibbling while I'm cooking, and I have had to force myself, like don't take bites of the ground beef until it's cooked, because I know, even though I like it. But I just don't understand why anybody would continue to eat it that way.
I guess this is a culture thing.
Yeah.
That's my guess. Sometimes our tongue or our habit of eating what kind of food has been trained while we are young.
Right. 61% is,
right.
I mean, that's high.
That's high. So
I don't know. Maybe science doesn't matter too.
Yeah, sometimes we give the scientific based recommendations, but are gonna follow it and not follow it. There are a lot of the other factors to trigger that.
Yeah,
Mhm.
it's interesting.
But at least we show something, and that could be the message delivered to the population. If somebody care, then you know if you reduce your consumption amount of the raw minced beef, then that can help a lot.
Yeah. So there's a lot of like the meat products. What about eggs and dairy? Did they
In this study, they didn't
were
look
just
at
looking at the meat.
Yeah, they only look at the meat. It's actually at least one more question at the end for us to think about. Other food can be the source, and we should broaden our research scope to include those food. Okay.
Can I ask a quick question on the same thing? I mean, it's more like a discussion point.
Yeah, sure.
On the previous slide, when we use our kill steps, which is usually cooking, if we are following the standards, you would expect to get 4 to 5 log inactivation for the pathogen. So I'm still surprised that the exposure risk is still so. I mean, it seems to be dropping about 3 to 4 log, but
yeah, if you prepare it right, if you cook it right, it should go down to nothing, right?
nothing, yeah,
If we cook it.
exactly. So why after adequate cooking, why is it still so high? That's
Oh, okay. So after cooking, why this is so high? I guess cross contamination contribute. But sometimes it is because maybe this model doesn't really fully represent what it should be. When you have the model, you have the data put into as the initial contamination level. Actually, I didn't show it here, but from the studies, it shows the initial contamination is important levels to have the influence about the final estimation of the exposure. But you can imagine that here we're actually comparing comparatively, make the comparison, but not really mainly focus on the absolute estimation. So here is more like I give you a rank and which is more important. And once we have the rank, then you know which food needs to be focused more and have a roughly estimation about the contribution percentage from different food commodity. I think this is the main message that they want to deliver. Again, it's like the model thing, and people very easy to argue that this is not accurate. If you have the garbage in, you have the garbage out. But depending on the purpose, there could be a preliminary step, and this could be further improved. And actually, for the people to do risk assessment, it is well recognized that doing a model like this to provide some recommendations in risk management is an iterative process. Let's have a starting point and continue to improve it. But again, here, to me, it's like, it's 10 to the 6 after this. To me, it's really, really high. So, no, it's because we also do risk assessment for beef and for chicken and for pork, and we have never had such a high estimation as this one. But this one has give you a relative rank. I think that's the most important message to be delivered.
Can I ask a quick follow-up? And maybe you mentioned it and I didn't catch it. Is it measuring the antimicrobial resistance risk from the entire microbiome or just the pathogenic microbes associated with the food product?
This is a very good question. So far for most of the quantitative microbial risk assessment, the data used is culture-based. So here they focus on the EEC, then the data is actually from the culture-based, that's the CFU or the presence and absence data of the EEC in the product. So it's not
resistant bacteria, but it may not be pathogen resistant bacteria. It's just presence of resistant bacteria, not necessarily presence of resistant bacteria is pathogenic.
Yeah, This is for the generic E. coli, so resistant generic E. coli. And so that could be the explanation why it is so high, but still it's still look a little bit high to me, because after cooking, we should expect a very high log reduction after cooking.
I mean, taking it back to like the clinical, going from food to clinical setting, right? So I go out to eat, I eat something, I'm immediately sick, right? Let's say, right? There's some bacteria like that did not sit well. And so you're immediately sick, but I mean, 9 times out of 10, or for me, 100% of the time, I don't go to the hospital for a follow-up. You got bacteria and you let your body deal with it. And so I don't know if that bacteria that got me sick, or I'm assuming it was the bacteria that got me sick when I went out to eat, was resistant or not. And so I'm wondering, just from like a clinical setting, you know, we saw the first thing where it shows like deaths from resistant bacteria. Do we have any sense of whether those deaths are through ingestion of food, or are they through like blood infection through a cut, or are they through like a surgery and like an infection? I don't know this information, but is that something that has been done in terms of like when we look at actually deaths from resistant infections? Do we know the exposure route and what percentage of that is, you know, eating meat?
You know, I think that's a very good question. I think that's everybody want to go the direction that want to go to. So it's hard to say. It's like that's starting from the infection in the human being, and then do the attribution to different possible routes, right? So it could be from the contamination in the hospital, or it could be from the food. But so far, I don't really notice any studies work that way, but there's a potential way. So all the studies here I showed is considered as a bottom-up approach. So bottom-up approach means you considered the hazard starting from the source. Like here is from the food and retail, and followed all the way to the human being exposure that is considered as a sink or receptor. Or there is another approach, it's called top-down approach. So if we have the information from the human being, if we have the information from different sources, then we can make the comparison and then start to make the attribution. It's actually, there are a lot of attribution model for foodborne pathogen illnesses, but not specifically for the resistant illnesses. So I think potentially there's a way to do that. It just needs to have the people working in the 2 different fields or multiple different fields collaborate together. I think one important factor is we need to have the funding allow us to work together. So far maybe CDC and FDA, they have the funding for the human being side and more our food side, and the USDA focus on the production side. But do they really have the funding that's put together that can have people from different areas work together? I think that could be that could be a trigger for promoting such research. Kind of happen. Yeah.
I have a question.
Yeah, sure.
You can hear me. Yeah, I was just thinking that it's not on this research, and I haven't read much about seafood, so I don't know why it's not. I don't know if it's like not given the same importance, or it's like a different category of risk or something. But I'd like to know a little bit more about that.
Seafood, actually, aquatic animals, they also use antibiotics, like how do I say?
Like farmed fish.
Right, from the fish and the shrimp. Yeah, they also use antibiotics. I think it's when people think about this, the major antibiotic use is actually go to the land animals or livestock. So most of the studies, they have work on meat and poultry, and even dairy, we don't have a lot of information from the dairy product. So seafood could be another one. But you can see this is a structure of a comparative risk or exposure assessment. Here is only consider several meat products and poultry products. But if we have the information from the seafood, from the plant-based food, from the dairy product, we can also include all of those into this comparison and make the relative attribution estimation. That is possible.
I feel like dairy would be much less likely source because dairy is processed before it gets to you. So it's been pasteurized.
As long as it's raw.
Yeah, well, raw milk is a whole other ballgame I don't understand. But,
I would say
other foods that come to you raw, then you are responsible for handling correctly to do that. So I don't know, dairy would seem like a lower risk.
Right.
And from what I've read with seafood, like oysters, those may be local, like here in Maryland, for example, but a lot of our seafood gets tested as it comes into the country because most of it's imported. And so we get tested at one point as it comes into the country, not every single piece, but there is some testing there. But again, a lot of it, it's coming from Vietnam, and we don't know the aquaculture, the practices and how much antibiotics, because it's this whole. Then you go into the global thing. I think Paul, you have something to say about that.
Actually, NAMS is doing a pilot study for seafood. So I'm doing sequencing for seafood to detect antimicrobial resistance.
Yeah. So that's something that's coming along. It hasn't historically been part of NARMS, and now you're bringing seafood into NARMS.
It's not there yet because we're just doing the pilot study, but it's going to be very soon on the website.
This is coming from Saul, one of our students who actually works at the FDA, and so he's saying that they're bringing that into their surveillance program.
Okay.
There's a short question. I still cannot understand this figure. You see, for the bill, the y-axis is total load, and the bill is after preparation is still like 6 logs. So why you say 0% to get? Like penalty?
Yeah, so and to repeat that, so if you look at veal as it goes down to the log 6, and also mutton, for example, is at log 6 the whole time. And so the question is, for this figure, that's not really 0%, right? Because it's still log 6.
No, this is not percent. It's actually the contamination level. So here's a total load. This is not 0%.
Yeah, so the total load, so mutton, for example, it looks like there's no mutton, but this is kind of the way this figure is presented. It's a little bit, I don't know the right word, but it seems like there's no mutton, but really it's just staying consistent at log 6. But again, this is model results. So this is not going out and this is the model outcome. So the model's having to stay at log 6. That's a good point. To me, it looks like there's mutton has nothing, but in reality, mutton's staying at log 6 in the model.
Generic E. coli, right? It's not pathogenic E. coli.
Generic and resistant E. coli.
Yeah.
Yes. Okay. So let's move on, and we can have. But I like such a good discussion. So, all right. So we can also do some more detailed quantitative microbial risk or exposure assessment to answer the 3 questions that we have talked about. And I have 2 examples to show this. And the first one is an example conducted in Canada. So what they're interested for the hazard is E. coli resistant to the 3rd generation cephalosporin and the tetracycline. And the food of interest is the ground beef. And they follow a chain starting from retail all the way to the fork for consumption. So you can see here it shows at a different step, different factors has been considered. For example, the time and the temperature during the transport from the store to the home, and at the home storage, are they stored in the refrigeration condition or freezers, and how long it has been stored in that condition, and how the time and the temperature affected the change in the bacterial population during that period. And also cooking is considered as the major thermal inactivation process And finally, they have the outcome measured as the exposure. So here's the outcome. Instead of how many CFU per serving can be expected, they quantify that the probability of consuming greater than 1 or to 10 to 100, all the way to 10 to the 6 CFU per serving of ground beef. And using that model, they also identify the critical control point throughout the chain that has been modeled. Okay, so here's the result. So in the baseline means under the current commonly used practice at retail, the transport, and the cooking or in the kitchen of the consumer. So they estimate the probability of exposed to or ingesting more than 10 CFU cephalosporin resistant E. coli is like 0.067%. So you can roughly interpret it as among 10,000 of servings, then you can expect 6.7 cases or servings having the cephalosporin resistant bacteria more than 10. So they also have the estimation for the tetracycline resistant E. coli. So you can see the number is much higher than the CREEC. It's actually not only in this study. We also do the study like this, and we constantly observe a consistent higher contamination level of tetracycline resistant bacteria compared to the third generation cephalosporin bacteria. And also they compared with a relatively higher threshold of contamination, and here it is the probability of exposed to this level. And then the bottom, that's the indication of some critical control point. And this is what is called a tornado chart. But they really flip over the tornado chart. If you have the longest bar at the top and the shortest bar at the bottom, it's really like the shape of a tornado. So this is how they generate it. So the tornado chart is generated based on if you change the values of a specific variables. So all of these variables, those are the input variable of the model. So if you change within this range, and how it's going to change the outcome. And here it shows you what is the association between you change the value of a cooking temperature and how it change the outcome. So it's give you a negative association of 0.86. So here, shows a strongest association between the cooking temperature and the exposure value compared to the other stochastic input variables, indicating if we want to control the exposure, then maybe control well of your cooking temperature. This is the most effective way to do that. And followed by that is the initial contamination of the bacteria in the raw product ranked as the second one. And also, they do a similar thing as the previous study. Along the chain of different steps, they estimate what is the level of contamination can be ended up in the raw or cooked product. So here, C3 is after storage, and C4 is after cooking. So you can see a very significant drop that is due to the cooking thermal activation. So this is another evidence to show that the cooking temperature or the cooking doneness is very important. In our group, we have done a similar project, and it shows we evaluated 4 different doneness levels. Well done, medium well, medium rare, and rare for ground beef. And if you want to contain a good texture or quality, then based on our study is medium rare is the level that you need to meet for ground beef. So the risk of exposure between the medium well and well done is very similar. But if you change medium well to medium rare, that can increase the exposure significantly. So that's the recommendation from our group.
Good to know. I don't have to order my hamburger medium or well done anymore. This is awful for me.
Okay.
So we are, just so you know, we are at almost 10 o'clock.
Right.
So we're over our class time. How much more do you have?
I think this is just one more example.
Okay. I guess I'm wondering, do we have a class period we could have Bing come back and cover this? We don't make you rush through it? Or we're filled up on our.
I think I can very quickly
Okay.
wrap it up.
Okay.
So this is another example. It's just a different type of antimicrobial bacteria, and also in different meat. And we also covered different procedures all the way from farm to the fork. So when we have the connections all the way from farm, then you can notice, okay, we can ask the question, if we change the use of antibiotics on farm, how is it going to have the impact in the public health? So here is the result. So here's the baseline. They have the risk level estimated. They have a different scenario analysis done to compare different potential interventions. So withdrawal preventive drugs, that is one of the intervention. And also they compared the use of antibiotics in human being and sanitation among the flocks on farm, and the cross-contamination, reduce the cross-contamination and processing, and the good temperature control at retail. So you can see here the percentages shows if you have a good control at those steps and how much risk can be reduced. So at the end, it's important to see, okay, to quantify and compare between different interventions, but also to see is actually all sectors along the chain needs to work collectively. Okay, so those are the examples I want to show you. And I have more questions. I just want to trigger more thinking from you guys. So at least several questions. And first is we don't have enough emphasis on the environmental pathways. And as I mentioned that there is some evaluation to compare different exposure pathways, including environmental pathway. But this is a focus on the pathogen, E. coli M157, but not specifically for the resistant bacteria. But we can imagine once we have the data for resistant bacteria from different sources and pathway, it is doable. So other questions is like, what is other type of food? We only have the animal-derived food, mainly meat and poultry, and how about the others? And the next one is, are we really taking a One Health approach? So everything here, we're talking about the risk, that's the public health risk. And how about the animal health? And there's no one criteria to incorporate that recipe in the discussion yet. And another thing is, now we have the shotgun whole genome sequencing, and how to use that information from that technology rather than only use the culture-based approach into the risk-based analysis. And the next one is, once we have some implementation, for example, in the United States, from 2017, we start to have the veterinary feed directive. And after several years, maybe we can have some evaluation about those interventions to see if it's really helpful for reduce the public health concern. And also, most of the risk assessment approach has been applied in developed countries that you can notice from the example. And I think it is a continuing work to increase the capacity building for the less developed countries to how to build the risk-based approach into their national safety management system. So I know I provide some readings, but here I provide more readings if you want to dig more into this topic.